The overall goal of the following experiment is to develop an easy and simple method to produce uniform three-dimensional neuroendocrine tumor spheroids and a simple method to process the histogel embedding for studying the histology of these spheroids. This is achieved by evenly coating 24-well plates with agarose on which human neuroendocrine tumor cells in a single-cell suspension are seated. The plate is then incubated on an orbital shaker in the incubator overnight at a speed lower than 40 rpm and then transferred to a stable shelf in the incubator. Spheroids will form by day 3 or 4, and their growth and integrity are monitored by phase contrast microscopy. In order to study their histology, the 3D spheroids are embedded in warmed histogel prior to routine paraffin processing. Results are obtained that show growth of neuroendocrine tumor 3D spheroids based on images taken during phase contrast microscopy. Histology of the 3D spheroids is analyzed by H&E and immunohistochemical staining of paraffin block sections of the 3D spheroids.
The main advantage of this technique over the existing method is that it's simple, easy, reliable, and cost-effective. This method can facilitate research in the drug discovery field by helping to quickly identify therapeutic drugs for the neuroendocrine tumor patients.
In general, individuals new to this method will struggle, initially at least, to grow uniformly shaped and sized 3D spheroids. The key to this is to ensure that your 24-well plates are evenly coated with agarose, and also to ensure that your single-cell suspensions are well dispersed and are accurate.
To understand the histology of the 3D spheroids, we need to perform paraffin processing and immunochemical staining. Histogel embedding of the 3D spheroids is the key step, which will be